to the NPTEL lecture series on animal physiology. So, we are in section 6 about the blood cells, immunity and the clotting. So, we initiated with the red blood cells in the first section, and then in the second section I bypassed the white blood cells and moved on to the platelets and the clotting factors. So, today what I will do, I will tell some of the basics. Now, since I have fairly good idea about what blood is, about some of the basics what whenever you go to a doctor and the doctor asks you to get a blood sample and a blood testing and what you see in the sheet. So, you know the doctor takes the blood or the compounder takes the blood and then it is being processed, and next evening or the same day or after some time you see a report. So, what that report really says and how you can interpret that report, this is very important for people to understand. Okay. This is a practical aspect of it. And next thing what we will be doing, I promised you that I will be covering how these different blood cells are formed, how this whole differentiation process, how a one single cell. So, let me rephrase this sentence. Essentially, blood is formed from one single form of, you can call it a pluripotent or a stem cell or something of that kind. So, from one source it all divides, some forms following a different pathway, red blood cells, some forms white blood cells, some form platelets, how that whole thing happens. Okay. And one more thing, well, in my last class while I was teaching you about the platelets, I made a small mistake, just correct it. I talked to you about the thrombocytes. Thrombocytes are not actually the platelets of the human being or mammal, it is for the non-mammalian. So, the thrombocytes are those. So, just a slight error. Basically, platelets are formed from megakaryocytes. I will come to that. Okay. Just correct that small error which I made in my previous class on the clotting aspect. Okay. So, talking about the blood, what a doctor does when doctor asks you to do a blood analysis. So, blood is a fluid that we all know now. By this time, we know that it is flowing all over the body, and fluid has lot of particles, and those particles could be cells. We have talked about red blood cells which are carrying the oxygen. Then we talked about the platelets. They are also small teeny tiny particle of 4 micron, kind of a major diameter, or it is kind of a triangular shape. So, you cannot call it a major diameter. It is kind of, if you take the maximum length of that triangle, the kind of the base of the triangle, then probably it will be around 4 micron, and those are non-nucleated. Same way the red blood cells are non-nucleated, they do not have any nucleus. Apart from, I talked about the platelets, that is where the thrombocytes come. Thrombocytes are the platelets of the non-mammalian system, which indeed have nucleus in them. Okay. So, we are not going to deal with those thrombocytes. Okay. We will be only dealing with the platelets which are in the human system. So, and then you have the third cell type, which is white blood cells, the cells for immunity, the macrophages. We talked about the macrophages while we talked about how the red blood cells which live their lifetime of say 120 days or 110 days kind of rejected out of the body, how they are being engulfed by the white blood cells in macrophages. So, the macrophages, if you do something called a blood smear. So, what essentially is blood smear is that, say for example, in my tip I just prick it and get a drop of blood and make a smear. So, you take that drop of blood and put it in a glass cover slip and you just spread it like this. Just like you have a drop of water, you do it like this, like look at my hand, and if this is the glass cover slip, you just smear it. And if you look at the smear, then there are several kind of, one second, excuse me. So, if you look at it under the microscope, you will see 3 distinct features. One, you will see, I told you about these platelet cells which do not have any nucleus. So, if you add any nuclear stain, you will not find any nuclear staining on them. Then you will find some cells which are circular biconcave kind of cells, which also do not have any nucleus. Those are your red blood cells, or there may be some red blood cell in the phase of formation which also does not have any nucleus. Yet you will find a kind of cell or a series of such cells which will have multiple nucleus connected to each other, which are multinucleated. Those multinucleated cells are your white blood cells. So, if I have to draw this, the classification of it, it will look something like this. On a blood smear, you will see. So, if this is your glass, then and if you are looking at a smear, you will see something like this, and then you will see something like this. It you will see a huge cells with something like this. Okay. Okay. So, now look at this, and going by the dimensions, if you look at it, these huge cells which I am now circling in red, these are your WBCs. These one which I put it in green are the platelets, and the one I am now putting in yellow are your RBCs. This is what you can see it under the microscope, okay, in a blood smear. Yet, these are the cellular component, but your blood is fluid, and what is the fluid component? So, if say just imagine, and in that smear, if you have a very high resolution microscope, you can see proteins. Just imagine, for the imaginary sake, you cannot really see it under like that with the known technologies. Then you will see a series of such proteins which are present there, and they are kind of floating in the fluid, which is mostly water. Okay. So, this watery fluid on which these proteins are suspended and the cell component are two different aspects. Okay. So, there are two different aspects of the blood. So, the blood essentially, if you have some way, say for example, you take the blood sample in a tube like this. Okay. Just draw it for your visualization. So, say for example, yeah, I give you a tube like that, and here you have filled with blood. Okay. So, this is all red is the blood. Okay. Okay. Now, if you if you take this blood and you spin it at certain RPM, okay, you put it in a centrifuge and spin it. After some time, what you will see after spin it down, this is what you are going to see. This is spinning or centrifugation is done to separate out components within a fluid. Okay. So, what you will essentially see, the fluid component remains at the top, and the cellular component or the solid component comes at the bottom. There will be separation, and separation will look pretty much like this. So, what we will say is lot of solid component here, and here you have a lot of fluid component. This fluid component which you separate out is called plasma, and this cellular component all is this, we can call it a cellular component, or it is technically it is called formed elements, and these formed elements are nothing but what I showed you here. These are the formed elements, WBCs, RBCs and platelets. So, now put the name for that, formed elements. And if I have to do a volume percentage ratio for this, these formed elements are around 37 to 54%. That is the constituent of the formed element, whereas the plasma component out here, this plasma component varies from 46 to 63%. Now, I talked to you about the different formed elements. Okay. Now, I will talk to you about the different proteins which are present there, and these proteins are essentially called plasma proteins. So, that formed the plasma component consists of protein elements. So, which you can imagine as if cell is a bigger particle, huge. So, protein is a micro nanoparticle. Okay. So, macroscopic particle at the micron range, and you have a nanoparticle. So, those nanoparticles remain suspended in the fluid, and the fluid consists of lot of electrolytes. So, plasma can be now put into two component, plasma protein component, plasma electrolyte component. Okay. So, that is what we will do. Now, let us classify the plasma. Okay, plasma into two parts. One part is called plasma proteins. This plasma protein, the major contribution is made by albumin. Albumin, all of you are aware of that in the, this is one of the major egg protein. Okay. You see that in the egg you have this albumin protein, bovine serum albumin and all these albumin. So, albumin is one of the major chunk, which is approximately almost I think around 60% of albumin. Apart from albumin, in the clotting I highlighted that you will be needing fibrinogen. Okay. So, fibrinogen is another component which is present there. Then let me enumerate them: albumin, which is approximately 60%. Then you have globulin proteins, hemoglobulins, that these are the globulin protein which constitute 35%. Then you have 4% of fibrinogen, and you have a bunch of enzyme, proenzyme, hormones, so which essentially falls under regulatory proteins, which is around less than 1%. Okay. So, this is pretty much the protein component which is involved in it. Okay. So, you have the huge chunk of the albumin, globulins, fibrinogen, and a series of hormones and proenzyme and everything. And this concentration of these different protein varies under different conditions of your body, different physiological status of your body, different pathological status of your body. So, by looking at the blood sample, analysis of the blood sample could tell with what kind of, technically speaking, one can predict cancer by very early diagnostics. But where is the problem? Why are we unable to detect it? This is the challenge. The reason we are unable to detect it lies in that less than 1% regulatory protein which I was telling you, all the hormones, enzymes, proenzymes and everything, because it is exceptionally challenging to, I should say, estimate proteins at a very low concentration. It is a big challenge. It is enormously big challenge, because and what level is higher than normal is another challenge to figure out, because whenever cancer or any kind of pathological situation happens, it is not that those proteins, they are already present in the body, but because of some XYZ wrong situation, their level either goes up or goes down. But how to make that demarcation? Because already these proteins are present in, proteins and enzymes are present in such a low concentration, and they work at it's a very low concentration chemistry, if you can call about it. It's a very femto, nano, femto, likewise that molarity, at that molarity these things are working. So, at that concentration it is really tough, and it is across the globe, whether you talk about NIH, United States, whether you talk about other similar organizations in Europe, back home in India, all the places still blood always remain one of the hot subject of research. Could we analyze all the component at far more early, before the we reach to a point, there is no point of no return, that time you cannot really, the patient will die of that problem. Okay. So, could we diagnose it? Is there a way to diagnose? Is there a way to diagnose cancer way early? Is there a way to diagnose some neurological disorder way early by just doing a blood sample? Could we predict? Do we have any mode? How low we can detect? It is all about the detection limit, and how accurately we can detect, but what is the level of accuracy? That is very important. So, that is why I highlighted this. That was the reason why I wanted to highlight this point. Those less than 1%, do not neglect them. They are exceptionally important, because that is where lies all your diagnostic kits, diagnostic tools and everything. Okay. Now, let us move on to the other component of the plasma. So, we talked about all these smaller particle component, which are at the nano, not only at the nano, I mean like at the nano size domain, Now, let us talk about the pure elemental composition in the form of the electrolytes. Okay. Let us get back to the slides and 1 second. Okay. The other solutes include your electrolytes, which of course, you have sodium, potassium, calcium, magnesium, chloride, bicarbonate, phosphate, sulfates. Likewise, 2 plus, 2 plus, minus. These are the series of electrolyte. Then you have the, in this fragment, you have the organic nutrients, which includes your ATP, cholesterol. So, this is another one, fatty acids. Then you have carbohydrate, you have amino acids likewise, and then you have the organic waste it carries, which includes your urea, uric acid, creatinine, bilirubin, ammonium ion. Now, having enumerated this, I will request all of you see whenever you get a blood profile check, what all the doctors can tell you. Doctors can tell you, get me the possibilities. Okay. The doctor can tell you, I wanted to see a blood cell profile. If the doctor asks for a blood cell profile, what are the possibilities? Either the RBC profile the doctor is asking, or a WBC profile doctor is asking, or a platelet profile doctor is asking. If the doctor is asking for a platelet profile, it means doctor has a suspicion about your clotting. Maybe there is excessive clotting, or there is a lesser clotting. Maybe something related to hemophilia or blockage in somewhere in the vessels. If the doctor talks about WBCs, then it has something to do with immunity. Fine. If the doctors talk to you about RBC, it has something to do with oxygen carrying capacity. Are you anemic, or you are hyperactive, or you are carrying more oxygen? Okay. So, look at it from one simple fluid. These are the 3 aspects. Now, if the doctor asks you, I want an uric acid profile, or I wanted to know what is the urea or ammonia, that essentially means doctor is doubting how your kidney function is, because urea and uric acid, this is all secreted by the through the kidney. Okay. Now, if the doctor asks, get me the potassium profile, this is very interesting. When the doctor asks for potassium profile, doctor may suspect that you may have some clotting problem, because if you remember, I was telling you that potassium plays a very critical role in all kind of clotting mechanism. Okay. If the doctor asks you that I want like profiles like electrolyte profile, the doctors may suspect that your electrolyte exchange is being compromised. Then doctor may ask you, I want a lipid profile. This is also doctors will ask you, just give a blood sample, we will get a lipid profile. Then the doctors are aware of, is your cholesterol is within control, or is the good cholesterol and proportion of good and the bad cholesterol, are they balanced in an optimal zone, or you are at the danger zone, you are at the susceptible zone for heart attacks or any kind of other disorders. Okay. They may ask for a lipid profile. So, look at it from one blood sample, you can predict almost, just randomly I could tell you at least 50 different situations which could happen in your body, because always realize whenever you study physiology, always it is a very holistic thing. This whole blood is flowing all over your body. It is not only picking up the element which it has to throw away from the body, the organic waste, it is also circulating the important compounds which the body needs for its growth, survival, and maintaining the homeostasis, as well as hemostasis. Hemostasis is the clotting, and homeostasis is the complete balance of the body. Okay. So, that is why blood analysis and blood sampling is so very important. doctor, first thing doctor will say, okay, or if the doctor suspect diabetes, I want the fasting sugar, non-fasting sugar. Okay. So try to look through any of those like the blood reports, and see on one column you will see what the analysis has told you, on the other column they will see this is the control, this is the maximum allowed, minimum allowed zone, and this is the normal zone, and then you have to correlate where your value falls in terms of the blood cell count or carbohydrate, urea, whatsoever. Then doctor may tell you, I want a bilirubin profile or the bilirubin concentration. That is where doctor is basically essentially indicating, doctor is suspecting you suffer from jaundice. So, these are some of the things which a blood analysis can tell you, and that is why after giving you some bit of introduction about the blood, red blood cells and the platelets, I came to this topic that will help you to appreciate why are we studying it in depth. Now, what I will do after covering this part of the basic the whole idea of blood testing and diagnostics and everything, what I will do, I will move on to the way the blood cells are formed. So, I will try to cover it in one slide with a pictorial representation, how within the bone marrow from one single cell type, all the different cell types are getting differentiated. The leftmost column we will be talking about the red blood cells, we will be talking about the white blood cells, then we will be talking about the platelets. Okay. So, in a one slide, this is how I am planning that will take care of all your doubts how these are being formed. Okay. So, let us back to the slide and talk about the origin of the or the basically the origin and differentiation of formed element. Okay. Origin and differentiation, the process by which a cell attains its final fate, differentiation of the formed elements. Okay. Okay. Let us get back to it. Fine. So, it starts with something called hemocytoblast. It is a huge cell though. This is the nucleus. Okay. Hemo means blood, hemocytoblast. This is the beginning point in the blood marrow, in the bone marrow. Okay. Hemocytoblast divides into two parts. So, hemocytoblast, part of it form lymphoid stem cells, lymphoid stem cell, and part of it form myeloid stem cells. Myeloid stem cell, I will just put SC henceforth, because that will help me easy. So, then these myeloid stem cells under the influence of factor called multi CSF, okay, they form certain elements called, and of course, there are a few other molecules which are involved in it, which is called erythropoietin, EPO, erythropoietin. Erythropoietin. Okay. The influence of EPO, some of these myeloid cells form the precursor cells for. So, this is the zone where we will be talking about all the RBC formation. So, let me put it in red. Okay. So, these are the progenitor cells for the RBC. This is an, let me put it like this, RBC progenitor cell. Okay. So, this is where the RBC progenitor cells are forming. And there is another series of progenitor cells from myeloid they are forming. I am putting them in green. Progenitor cells. These progenitor cells form something called megakaryocyte, and these megakaryocytes are cells like this, huge cells like this. Okay. So, what I will do before I come back to the right side, like all the lymphoid stem cells and all other things, what I will do, I will just talk about these two elements. I am just putting them in, these two elements we will talk about first, otherwise it will become very complex. Okay. We will talk about these two elements, the what is happening here. So, let us first of all talk about what is happening to the RBC progenitor cells. Okay. So, we start with after the, so the first series of cells are called proerythroblast. This proerythroblast reached into erythroblast stage. Erythroblast is the other name for the RBC, erythroblast. Okay. And from here it forms something called reticulocyte, and reticulocyte is the phase when it starts losing the nucleus, which you remember I was showing in the first class, how they are losing the nucleus because of 15-lipoxygenase activity. So, this is the zone where the reticulocyte is the stage when from erythroblast to reticulocyte formation. This is the stage when there is a sharp peak within the cytoplasm of 15-lipoxygenase enzyme, which I have already discussed in the first class. And because of that 15-lipoxygenase enzyme, all the different cell organelles and the nucleus completely gets damaged. So, the cell is only filled with hemoglobin molecule, which helps for rest of its life of 120 days to carry oxygen. Okay. From reticulocytes, what we get is essentially the RBCs. Okay. So, this is the pathway which is followed, which started with, if I have to summarize, it started with hemocytoblast, then you have myeloid stem cells. From myeloid stem cells, because of the influence of erythropoietin, some of these progenitor cells, these are the progenitor cells, form proerythroblast, and proerythroblast become erythroblast, and then erythroblast becomes reticulocyte, and that becomes RBC. Okay. So, we are done with one set of reactions which leads to the formation of the RBCs. Now, what we will do? We will talk about the second one, which is the formation of the platelets or the clotting elements of the body. Okay, let us get back to that. So, what is happening here? Okay. So, same again, starting with hemocytoblast to myeloid stem cell. The myeloid stem cells, under the action of multi-CSF, which is multi-CSF stands for colony stimulating factor, okay. These myeloid stem cells from progenitor cell for which are destined to become platelets. Okay, progenitor cells for platelets, and then they form a structure, huge structure called megakaryocytes. Of course, the megakaryocyte has nucleus, mind it. Okay, megakaryocyte, and this megakaryocyte then divides into small cells, the platelets. So, this is the second route, by the stimulation of the myeloid stem cells by colony stimulating factor. These cells form a huge cells called megakaryocyte, then megakaryocytes started fragmenting. They basically, as I told you, platelets are fragments of a bigger cell. They do not have any nucleus, at least in the mammalian system. In the non-mammalian, of course, in the thrombocytes they do indeed have, but that is a totally different mechanism, and we are not going to discuss this here. So, these fragments of the cells form the platelets. Now, we come to the much bigger and the tougher one, which is how our immune cells are formed, because as of now we bypassed all that. Okay. We talked about RBCs, we talked about the platelets. Now, let us get back to that whole hemocytoblast, and we talked about the lymphoid stem cells, and I have not talked about it. Now, I am going to talk about part of the lymphoid stem cells and part of the myeloid stem cells, and how they are leading to the formation of the different white blood cells, which are almost 5 types, okay, eosinophil, basophil, lymphocyte, leukocyte, likewise. Okay. Let us get into that. So, let us move on to the next slide. So, again we start with hemocytoblast. Now, let me use some other color. Okay. Hemocytoblast. So, I showed you the one classification on the one division on that side, because they form lymphoid stem cell. Okay. And lymphoid stem cells form something called lymphoblast, and lymphoblast form something called prolymphocyte, and prolymphocyte forms lymphocytes. This is one route. There are multiple routes, which I am coming to in the next. So, this is where all the lymphocytes are formed. So, white blood cells could be classified into 2 groups. One group which has the lymphoid lineage, they form from hemocytoblast to lymphoid cells, and then through a cascade of lymphoid stem cells to lymphoblast to prolymphoblast, and reaching all the way to lymphocytes. And the lymphocytes have further classification, which I will be coming soon. Lymphocytes could be B cell, T cell, likewise. I am not getting into that at this point. Okay. So, this is one route, which is the lymphoid route, but I showed you that this is another route, which is myeloid route. What is happening in the myeloid route? What they are responsible for? Because already we have seen when hemocytoblast is divided into myeloid route, it leads to the formation of RBCs and the platelet. Okay. Yet a part of this myeloid stem cells do form other components of the white blood cells. Okay. So, we will now discuss the second route. So, hemocytoblast to what I will do, I will just pick up another color, so that will make your life slightly easier to understand. Okay. We have already talked about 2 routes, where hemocytoblast. Okay, let's do it like this. This is the wrong way to show it. Okay, fine. Here let us put the myeloid stem cells. Myeloid stem cells. Okay. From myeloid stem cells, I talked to you that through erythropoietin something is from RBCs. Okay. This part is all taken care. Okay. Then through colony stimulating factor, some of the cells are forming platelets. Okay. This part is all done. So, we are not going to deal with this part. Now, we will talk about what is happening to the myeloid stem cells, then out here. Okay. So, some of these myeloid stem cells are under the influence of GM and CSF, colony stimulating factors, to progenitor cells, which are destined to become white blood cells, progenitor cells for WBC, white blood cells. Okay. These progenitor cells then divide into 2 parts. These are called one part, they all fall under one part, which are called blast cells. These are called myeloblast. Okay. This myeloblast further form 3 kinds of myelocytes. Okay. These myelocytes then form something called band cells, and these band cells form 3 different white blood cells. One is called basophil, the other one is called eosinophil, third one is called neutrophil. Whereas, there is another route I showed you, there is something which form, here is from myoblast, here is from monoblast. This monoblast form thing called promonocyte, and promonocyte form monocyte. So, this is the other route, which is of course, let me just separate it out. At this point, this is the demarcation taking place. Okay. This is where the 2 different cell types are getting separated from here. This is one type, one separation, this is the second separation, and this is the second route. So, in sum total, you have basophil. So, if you look at it, what are the different kind of white blood cells? Okay. You have basophil, you have eosinophil, you have neutrophil, you have monocyte, which are coming from myeloid lineage, and you have lymphocytes, which is coming from lymphoid lineage. So, in sum total, there are 5 different kind of WBCs. Now, if you, in the light of this information, if you look at the blood chart, when the doctor asks you that get me the blood report, they will say eosinophil concentration is higher. Generally, whenever you have persistent sneezing or something, doctor will say, oh, you are suffering from eosinophilia. What does that mean? That means your eosinophil cell number has gone up. These eosinophil, what you see, which I draw, which is coming from myeloblast and myelocytes and the band cells. Okay. And if you look under the microscope, how they look like, they look something like this. All these different, now I am talking. So, basophil looks like this. There are a lot of pigments all over the place. Okay. And if you look at eosinophil cells will look like this, something like this. So, this is eosinophil, this is, of course, they have to be put different kind of dyes in order to color them. Okay. Then, if you look at the neutrophil, their sizes are fairly close to each other. Okay. I mean, so these are all multinucleated cells. Okay. So, this is called neutrophil. Then you have monocytes, which is more compact kind of, this is monocyte, and of course, lymphocytes also is slightly smaller though, and like this, which is lymphocyte. Okay. So, coming back. So, if I had to summarize it, so we talk about hemocytoblast, myeloid origin, hemocytoblast divided into myeloid lineage, lymphoid lineage. Myeloid lineage leads to the formation of RBCs, platelets, and basophil, eosinophil, neutrophil, monocyte, which are all clubbed under the category of white blood cells. And there is another set of white blood cells which are of lymphoid origin, which are formed by from the lymphoid lineage, which are called lymphocytes, and lymphocytes are further divided. Okay. What we will do now, after telling you this, I want you guys to visualize whenever you heard about somebody has a blood cancer, what really that means. Now, in the light of this whole differentiation what I highlighted in the bone marrow, what is happening from hemocytoblast, think of it how tightly this whole process is regulated. Each cell number has to be right. If it is wrong, then there is a problem. Each cell number has to be tweaked in such a way that if something goes bad, you may suffer from hemophilia, you may suffer from clotting disorder, you may suffer from anemia, the oxygen, or you may suffer from, or you may have higher oxygen carrying capacity if you are fortunate. Okay. Then you will be very hyperactive. You may have more number of you may be immune compromised, AIDS. Okay. You are immune compromised. So, immune compromisation means you have problem with your white blood cells. So, in the light of this whole differentiation process, from the hemocytoblast, this whole differentiation of blood cells is forming all throughout your life within the bone marrow, that there is enormous research is taking place across the world to understand this process, because this is that zone where lies some of our answer to cancer. Because if you have to understand cancer, this is where we have to hit upon. One of the, of course, related to the blood cancer, this is where it is, because this whole differentiation is very, very tightly regulated. I mean, like, there is one flaw from this, the person suffers. Okay. So, that was the sole reason why I took this whole class single, exclusively to give you, in the light of this, when you look at the whole thing, that will make more sense to you than looking them at small fragments, bits and pieces. So, that is why the blood analysis is so very important, that what are you analyzing in the form of blood. That is the sole reason why I just started with your RBCs, talked to you about the clotting, and then came to and introduce you to the whole lineage of WBCs, how they are formed. It is from the same cell. So, what regulates? Some of the open ended questions which remain, which are to be answered by mankind, is that what is so unique from the same population of cell, some becomes RBCs, some became neutrophil, or some became platelets. Could we? So, the future of tissue engineering, future of differentiation, development biology is that could I pick up any cell, any blood cell, or those kind of progenitor cells like hemocytoblast, and say, okay, I need more number of platelets, or I need more number of RBCs, and could he put it back? That is the translational medicine where it is all heading. Could he do that? That is the future where it lies. Could we control the differentiation and the development of these different kind of formed elements, and if we could, and how we could. This is where lies some of the very, very challenging questions of next century or this century. As a matter of fact, that could we really do that? Could we cure a patient of blood cancer? That we know, don't worry, we can take out a cell, or there is another way. Could we take any kind of stem cells and transform them into blood cells or any other cell type? So, these are some of the very open ended questions which for which people are striving all over the world. The scientists are working day and night to figure out some of these answers, because it is very easy to see the chart, but the fact is physiology is far beyond those charts. How really to do that? Yeah, of course, this is only bits and pieces. I mean, if you go back to this chart, what I was trying to draw and show you guys. Like, think of it, there are so many factors which are involved, about which you have no idea, which are really leading to this. What is leading to this? What are the signal? So, that is where lies a lot of unanswered questions out here, or say for example, here proerythroblast, erythroblast, reticulocyte. There are so many factors. What are those factors? And those are happening those are very small molecules. They are expressed for a moment, and that is it. They moved out. Okay. How we can control differentiation? These are some of the questions, and that is why that whole chart I wish you guys to redraw that chart in a bigger paper. So, that because there is a size and limitations of this screen, that I cannot draw everything unless I am in a blackboard where I can have the whole end. Okay. So, I want you guys to redraw this whole thing, what I have covered now, as of now. That will give you a kind of understanding how this whole process is taking place. So, what I will do now, probably in the next class, what I will be doing is that we will be talking about the function of the different WBCs, and we will talk about a little bit about the immunity and a little bit about the RH factor. There are a couple of things which I have not touched. So, let me tell you what I will be touching in the next class. I will be talking about the blood groupings. That you guys have heard this A plus, B plus, O, and what is blood grouping really? That is one aspect what I am going to touch. Second, I am going to touch about a little bit of immunity. Third, I am going to touch about all the different kind of WBCs which are present. So, all the different kind of WBCs, blood grouping, and bit of an RH factor, RH positive, RH negative, and how that influences the pregnancy. We will talk about all these things and a little bit of immunity, and that is where we will close in with this section of blood cells, immunity, and clotting. Thanks for your attention.